Hi, my name is Emily, and today I'm going to be talking about sourdough. And you may be thinking, why are you going to talk about sourdough? One, because it's delicious and amazing. And two, because it's the result of a huge and very diverse population and community of microorganisms that interact with each other and with their environment to make the amazing sourdough that we all know and love. I'm going to talk about those interactions. Let's go to the thought board to show you what I'm talking about. All right, so welcome to our thought board where we will be making sourdough. To make sourdough, you only need two ingredients. First, you're going to need some flour, just some regular old bread flour. And secondly, you guessed it, we need some water. And when we mix these two together, then the magic happens. Our sourdough starter will start to rise and get bubbles. This is due to three magical microorganisms, the first one being yeast, particularly those belonging to the Saccharomyces and Candida subspecies that produce CO2. The second one are lactic acid bacteria, mainly in the Lactobacillus subspecies, which produce lactic acid when they break down sugars. Finally, our third one is acetic acid bacteria, particularly the order of Rhodospirales, which produce acetic acid when they break down sugars. Together, these three magical microorganism types help to produce the flavor of the sourdough, as well as CO2, which helps the sourdough starter rise and forms bubbles for the texture. That gives us one happy loaf of sourdough. Now that we have a background on sourdough, we can move on to our research question, which is, how does stress impact the microbial population of a sourdough starter, particularly salt stress? So we're going to add varying levels of salt to sourdough starters and see how that impacts our microbial population. And you might be asking, how will we know how it impacts the microbial population? Well, I'm glad you asked, because we will be using metagenomics. And what that means is that we will be taking the genetic information from the entire community and sequencing it and producing a large data set that will tell us all of the different kinds of microbes that are in our sourdough starter. Let's jump over so I can show you what we did and how we sequenced the information. So this is where the magic happens. In this course, we use KBase, which helps us to sequence all of the DNA data that we had using Sanger and Illumina sequencing. And it gives us a bunch of information like total sequences, sequence lengths, and contigs. And you might be wondering, Emily, what do you mean by a contig? Let's jump back to the thought board and I'll show you what we were talking about. All right, so when you sequence a genome, it takes a long time because it's a lot of base pairs to read. So to speed up the process, we can break up the reads into short little segments, and you can see all of the little gaps. But if you combine all of those little reads, you get a contig, which removes all of those gaps. Then we can use assembly and assemble those two contigs together, and that removes that gap in the middle. And that makes a continuous genome, which we can compare to reference genomes to identify a microorganism. All right, so now that we know a little bit about genomics and about sourdough, let's go into the results that we have from our experiment. All right, so here are the results from the first sample. You can see here is our control sample. And in our control sample, you can see that our most common yeast is Saccharomyces leadwicki. And that makes up a large portion of our metagenome. And you can also see we have the presence of Klebsiella pneumoniae, which, while not being one of our prototypical lactobacillus bacteria, does occasionally produce lactic acid as a byproduct. And as we go further down, we also see that we do have some lactobacillus lower down, and they don't take up quite as much space on our chart, which means that they are not quite in the high numbers as these top two the Klebsiella pneumoniae and the Candidatus cryptobacter tencongensis. Now, this second chart shows the first treatment of salt, and you can see that the salt does change the population of microbes in our sourdough starter. So one, you can see that the Candidatus cryptobacter tencongensis population has increased immensely and pretty much doubled in size when compared to the control, while you can see that the Klebsiella pneumoniae stays relatively the same. And our Sarcomycotes ludwigi reduces a little bit, but is still our most populous yeast. And then the rest of our bacteria stay somewhat consistent as we go down the line. When we compare this to our species tree, we can see that the metagenomes that we sequenced are not as related to the species given in our species trees as we would think that they would be. You can see that because they're sort of ancillary and that the control and SALT1 are not connected to any of these lactobacillus species, that they are not closely related. And this SALT2 sample is more related to the others, but also not as closely related as many of these other species. This could be the result of bacteria or other microbes coming from lab workers' hands or from the specific environment in which these sourdough starters grow. And because sourdough starters are unique and diverse, each sourdough starter is unique and has its own bacteria. So here are samples from later experiments. Our control 2 is seen here, and we can see that we still have a lot of Klebsiella pneumoniae, and that is our most populous bacteria. We can also see that we have some Acinetobacter baumannii, and our most populous yeast is Sarcomycotes ludwigi, just like it was in our old control. When we look at our new salt solution, our new salt sample rather, we can see that there is again a drastic change. We have slightly less Klebsiella pneumoniae, but we have this new bacterium, Chlamydia abortus, which was not present in the control sample, as well as slightly fewer numbers of the Acinetobacter baumannii and the Sarcomycotes ludwigi yeast. And that is interesting to see how the salt concentration between these two impact the species that are available. You can also see that some of these microbes further down, there are a lot more found in our control than there are shown in our salt. So you can see that the diversity has changed and that we have fewer lines at the bottom. And also that we are missing some of these other lines in the salt concentration. And these two brown lines are actually two lactobacillus bacteria, which is interesting to see how the introduction of salt into this control actually got rid of some of our lactobacillus. And when we compare that to our species tree, we can see that these two metagenomes for our salt are not closely related to any of the other genomes that we have on reference. And that's because if we look back to that, we see that these two lactobacillus are no longer present in our salt sample. And so this makes sense why these are not particularly related to these others in the tree. But we do see that our control is quite closely related to the Lactobacillus psittaci and Lactobacillus jensenii bacteria species. And this also makes sense because we saw that we had Lactobacillus species present in our metagenome. And therefore, they're more closely related to all of these others. When we compare the two data sets, we can see the change in between the different controls and also in between the impacts that occur due to the salt. So we can see that our most populous bacteria here is the Candidatus cryptobacter tencongensis, but across the board, Klebsiella pneumoniae was very common in our controls, and that Sarcomycotes ludwigi is our most common yeast between both samples. We also see that there is a change in the diversity in between both of our controls and then the salt. So we can see that there are a ton more colors and lines present in our control groups, and that when we look at that compared to the salt, that we see fewer lines, especially when we look at the experimental sample 2, in which more salt was added. We see less diversity. We also see that Lactobacillus species are not the most populous in our samples, which is not expected. Normally, Lactobacillus are quite popular, large contributing bacteria to our sourdough starter. So it is interesting to see that we have many other microorganisms that are not the first microorganisms that would come to mind when we think of sourdough starters. It is also interesting to see microorganisms that are present in our salt samples that were not present in our control samples, like the Chlamydia abortus being a very popular bacteria in our salt sample 2, but not being present in any of the other samples. When we compare the two species trees, we see that control 2 is actually more closely related than any of the other samples. And that makes sense because we had more Lactobacillus present in that sample. And so it fits better into this tree. Whereas the other samples didn't have a lot of Lactobacillus bacteria present, and so they are not as closely related and are shown on the outsides of these trees. So now that we have all of this information that we gathered in this experiment, where do we go from here? There are many other experiments that analyze the difference between a typical sourdough starter and the microorganisms present in it, as well as the microorganisms present on the hands of people that work with the sourdough starter, and compare which microorganisms are more present in the sourdough starter and which ones seem to originate from the human's hands. I think that would be an interesting direction into which to further analyze these results, since we had some microorganisms present that were not the most typical microorganisms found in sourdough starters. It would also be interesting to start a new mother batch of sourdough starter to see how the control population differs, and to experiment on different stresses other than salt, such as not enough sugar or varying temperatures, to see how those impact the stress and the reaction of the microorganisms in that population. Some of these results were surprising, such as the presence of new bacteria in the salt samples when compared to the control samples. To me, that was probably the most interesting part of this experiment. It would be great to replicate the experiment over and over again to see if there is a pattern present in understanding which bacteria and which other microorganisms are present over time and throughout stresses. I think through repetition of experiments, we would be able to differentiate between native species of microorganisms in sourdough versus species present that are native to human hands or instruments in the lab, so that we could understand if the sourdough had microorganisms in it that were not originally native to the sourdough. And I think that would also help us to understand what changes when we add stress to the sourdough. This is new research, and sourdough populations of microorganisms in sourdough are not yet fully established. So I think just being a part of this research and doing it repetitively to find a pattern in the results would help to give us a better understanding of how microorganisms in sourdough starters react to salt stress. Then with that information, we can generalize it and see where it goes and see how other environmental factors affect the interactions between and among microorganisms and how they survive in a sourdough starter. Thank you very much for watching this video. I appreciate it.